Look at this mapping of medicinal plants and fungi to their complex chemical structures. Roughly 50% of modern pharmaceutical drugs are derived from these types of natural origins. This diagram illustrates how those biological sources extend beyond plants and fungi. Marine environments provide a massive pharmacological resource, offering compounds that researchers use to develop anti-cancer and antiviral agents. Because producing organisms naturally develop these secondary metabolites to defend themselves against pathogens, nature acts as a highly evolved chemical laboratory. That makes it a logical starting point for discovering new therapeutic agents. Treating viral infections presents a unique challenge. Unlike bacteria, viruses cannot survive on their own. They rely entirely on hijacking healthy host cells, injecting their genetic material, and forcing the host to multiply the virus. Since they are so tightly bound to the host's own biology, finding a treatment that kills the virus without killing the host cell is difficult. Researchers ask, how do we determine if a new natural extract can actually neutralize these viruses? To answer that question, laboratories rely on a highly specific procedure called the virus inactivation assay. The core purpose of this assay is to test a direct physical interaction. We want to prove definitively whether a specific chemical extract can destroy a virus particle entirely on its own upon direct contact. This flowchart maps out the complete roadmap for the virus inactivation assay. Step 1 begins here, at the top left. We take a standardized sample of the virus, 10 to the 5th plaque-forming units, and mix it directly into a test tube with our natural extract. The logic here is straightforward. We need to expose the virus to the potential medication, while the viral particles are freely floating in the solution, completely independent of any host cells. Once mixed, we move to step 2. We place this solution into an incubator set to exactly 37 degrees Celsius for 2 hours. 37 degrees Celsius perfectly mimics human body temperature. This 2-hour window provides the exact chemical conditions and time required for the active compounds in the extract to potentially break down the viral envelope. Following the incubation, we reach step 3. We must immediately perform a series of high dilutions on the virus extract mixture. This prevents a common laboratory trap. Many natural plant extracts, while highly effective against viruses, can also be toxic to the delicate laboratory cell cultures we use to read our results. If we skip the dilution and pour the potent raw extract directly onto our assay plate, it will indiscriminately kill the host cells. That ruins the entire experiment, because we won't know if the cells died from the virus or the treatment itself. The high dilution step solves this. It dilutes the extract until it is no longer toxic to the host cells, while leaving enough of the target viral particles in the liquid for us to observe. Now we can proceed to step 4. We take this diluted and hopefully inactivated viral mixture and add it onto a plate containing a healthy, confluent layer of vero cells. Vero cells are a specific lineage of cells grown in laboratories to culture viruses. They act as biological host indicators. If live, infectious viruses survived their time with the extract, the vero cells will show us. For step 5, we cover the infected vero cells with a semi-solid overlay and return the plate to the incubator for several days. Inside the incubator, any surviving viruses will enter a cell, multiply, and burst out. Because of the semi-solid overlay we added, the newly released viruses can't float away. They are forced to infect and kill only their immediate neighboring cells, repeating the cycle. This localized spread creates a small, visible dead zone in the cell layer, which we call a plaque. Every plaque is physical proof that a viral particle survived the initial treatment. Looking at the final step shown in the bottom right of the diagram, we take our plates out and count those plaques. We then compare our treated plate against a standard control plate that received the virus but no extract. A control plate will be riddled with dozens of plaques. A successful treated plate, however, will show a mostly solid, healthy cell sheet with very few dead zones. We divide the number of plaques on the treated plate by the number on the control plate. This simple math provides the exact percentage of viral inactivation. If the plaque count drops from 100 on the control to just 2 on the test plate, you have 98% inactivation. That dramatically lower number mathematically proves the natural extract acts as a direct, effective antiviral. So far, we've successfully prepared the virus, exposed it to a natural extract, protected our host indicator cells from toxicity, and quantified the extract's destructive power. But this raises a critical scientific question. What if you test a promising plant extract and your treated plate ends up with just as many plaques as the control? A failure in the direct inactivation assay does not mean the extract is useless. It might simply neutralize the virus through a completely different biological mechanism. This flowchart introduces those alternatives. The active compounds might not destroy a free-floating virus, but they might prevent the virus from attaching to a cell membrane in the first place, or block it from injecting its genetic material. Determining antiviral efficacy isn't a single test, it's a multi-step investigative process. To find out exactly how a natural drug protects a cell, we have to move on to time of addition and entry assays in the next phase of research.